Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show. Today, bringing you another really awesome guest involved in science and technology, creating a better tomorrow for all of us. And today, we're going to be getting back into a really interesting topic we haven't touched on actually in a couple of years, and namely that of bacteriophage, or more informally known as phage, these really cool viruses that like to infect and replicate and kill all sorts of bacteria. And as there's quite a few, something like 10 to the 31st power or so of them on this planet. We've only really begun to tap their potential in human health care. Today, we have the honor of being joined on the show by Dr. Jessica Sacher. She is the co-founder of the Phage Directory. This is a really interesting global network of phage researchers from over 80 countries around the world, where she directs their sourcing, their community building efforts, ultimately coordinating something like 50 different phage finding efforts with a network of something like 1,300 members of the global phage community. She's also a staff scientist at Stanford, University School of Medicine, where she leads strategy and lays groundwork for new phage-based therapeutic development and delivery at Stanford. And they're developing really interesting manufacturing processes and quality control methods to create this clinical grade pipeline. She was previously a member of Professor Jon Iredell's group in Sydney, Australia, as a postdoc research scientist for Phage Australia. And there she was developing and streamlining Phage Australia's selection, biobanking, manufacturing processes. She did a Ph.D. in Microbiology and Biotech from University of Alberta, Canada. She's also a founder, editor, writer of Capsid Tail. That is a really interesting phage community newsletter where she weekly reports on bacteriophage and news and research, and also it's a pretty cool podcast as well associated with that. A lot of really cool topics to get into today. I'm very honored to have her. Dr. Jessica Sacher, thank you so much for taking the time on the show.
Thank you so much. That's so kind of an intro.
No, it's a cool background, and again a really cool topic, and I wish I did more on bacteriophage, but I'm really looking forward to this episode and where we're going to be going. Jessica, I'd like to start off just by handing things over to you for a little bit to talk a little bit about your background and how you got involved in this space. I was taking a look back at your dissertation, which focused on Campylobacter and this really interesting phage that you were studying that it's called NCTC 12673, which it came up with an interesting property that it liked to mess around with the flagella and the flagella-related glycans that Campylobacter like to hang out and float around with, and then kind of cool mechanism there in inhibiting that bacteria. But I'd love to hear your background story and a little bit of what got you focused on the space.
Yeah. Wow. No one's brought up NCTC 12673 in years. and unfortunately named because it's just the name that it's been assigned. I never got to give it a cool name because we weren't doing that at the time. But yeah, I got into microbiology because I wanted to be a veterinarian. And then so I took science degree, and then I realized didn't want to be a vet because it was too sad. I was volunteering in a clinic and just sadness every day. And I found microbiology, I think just through one of my undergrad classes, and couldn't believe that there was these unseen microbes just living everywhere, controlling everything. And I think like the lecture that captured me was the one, it was around 2008 or so, like the microbiome, the gut microbiome was just becoming kind of something talked about. And they had transplanted the gut microbes from obese mice into thin mice and made them obese. And I was like, what? Like, how are people not talking about bacteria and how much power they have, like this whole extra organ? So I kind of just stopped applying to vet schools, I think, around and then, and I started just trying to find research opportunities. And I was a big Pokemon fan when I was a kid. So
Nice.
microbes just feels like, they're all out there. You have to collect them all, and they each have different powers. And I think that thread has remained true because as I found phages, just as the professor that I really liked, Christine Szymanski, she was studying phages as well in her lab. And phages were even more Pokemon-like because you could use them in battle
Yep.
to kind of delete whatever you wanted. And so, yeah, I've just been pursuing curiosities basically in that realm and just always kind of surprised when things are so powerful but not used yet. And just the quest to kind of get them useful. And so that's kind of what led me, like I was finishing up my PhD. This is around 2017 at the time, and wondering what I should do next, wondering if I should do academic research as a career, and feeling like that was just too slow and too, I don't know, too hard to win. Like it just seems like you're running on a treadmill to try and publish papers. And and if you don't do that, you can't keep playing the game. And yet the papers weren't really the thing that felt like impact. So that's about when I met my partner Jon, who I at the time knew from swing dancing, and just kind of he was really cool. We were having coffee, and that's when we like figured out that people were using Twitter to actually find phages to help real patients. And so that was like the birth of Phage Directory because Jan said, like, why is your field using this like ill-equipped tool and just hoping somebody sees a tweet? Like we could build something that actually makes these things useful for the clinic. Like, why aren't we building this? And
Mm-hmm.
so, yeah, that's kind of like that turned everything for me kind of a career turning point, I would say, because I
Yeah.
was like, why wouldn't we? Why wouldn't we build this?
Exactly, exactly. And yeah, that point about, the things that we are unused. And, I'm a strong proponent that, nature, and in this case, viruses, have a lot to teach us. They've been around longer than the bacteria, which have their leg up and their advantages over us. And, we got to sometimes look to the other species that are good at dealing with them. So I very much enjoyed the concept behind sort of phage, and it's just so interesting that I had the opportunity to host folks like Steffanie Strathdee and Bob Schooley from down the road from you in San Diego, and we have something here in the sense that a concept of, hey, let's use this virus to kill this bacteria that goes back, 100 years now or so. I think, used as therapeutics back in the 1910s and 1920s. Then this penicillin thing came along and kind of upheaved everything. But nonetheless, certain corners of the world, it took off a little bit. Could you start out a little bit by giving us a bit of the history of sort of how things went, let's say last century, and sort of why it didn't take off, and as I hope it will be taking off now. And give us a little history, if you would.
Yeah. Yeah. So phages are phage therapy. It's all this weirdly old but still new technology. So we've been in this weird place, I feel like, for a while. So we passed the 100-year anniversary a few years ago, and everybody celebrated it. And it's so confusing. Like, why do we have something for 100 years and we're still not using it in the mainstream in the clinic? It does it either not work and therefore we should abandon it, or does it work? And then why can something work but still be not really used? So the way that I think about the history is, yeah, back in the, before the 20s, 1914, 15, that's when phages were discovered. And people at that time, like we didn't know about DNA. We didn't really know about, you know, viruses. Like we couldn't easily see microbes. And so what phages were just like a little clear zone on a petri dish where bacteria were growing. So kind of like your moldy food in the fridge. And if you saw like a piece where the mold was gone, like in a suspicious like circle, and you saw a bunch of those, like that's what it would have felt like to have discovered phages. And so they didn't know what they were, but they knew that there was some entity that was invisible that was killing bacteria. And you could kind of pick up a little bit of it and put it in a new vial, and that vial would contain that thing. And so they knew it was like a discrete element that had this killing power. So yeah, that's when they're discovered. And they quickly started being used therapeutically way before there was knowledge of what they were. And people were putting them in rivers. And there's like all kinds of cool historical figures like Felix Durrell, who is like this renegade self-taught microbiologist who was one of the co-discoverers of phage. And so he was, getting, I think, calls from all these governments to like come and help deploy phages after a while. And I'm not the historian that knows the exact chronology, but like it was in this time he was thinking, why don't we use these things that kill bacteria to get rid of the bacteria that are killing us? And so they treated like kids, and yeah, there was a lot of treatment going on. I think chickens as well. And these things were kind of like sometimes working and sometimes having amazing effects, like putting them in one side of like in the river of this one town in India, but like only on one side of it. So they could see like kind of a control group, like what was the effect of diarrhea in all these people. And it was helping, but we didn't have the same requirements or bar for proof at that time. Of course, it took a while for us to develop our regulatory system. So yeah, that's what it was like. And there was a lot of controversy around, I guess, around this time. So I guess Stalin was really into phages, and so he kind of was, wanting Felix Durrell to be able to develop this. And so there was this. This is the birth of the Eliava Center for Phage Research and Therapy, and that's in Tbilisi, Georgia, the country of Georgia. And so that was, like this, it became this clinic, and it's still around. It's been there for 90 years. So, and there was, all these, the story of the whole Soviet Union phage, that whole story, there's books written about it that are awesome, that like if people are curious, it's pretty crazy. But anyway, so phage is just kind of like, it kept going forward because it was working. It must have been working enough at the time that people were feeling like it wasn't useless. But as we learned about in like the 50s what DNA was, and you could start to figure out the sequence of pieces of DNA, people were actually using phages a ton in the from that point on in molecular biology, just to figure out what is DNA. Like it's because of phages that we even know what the heritable thing is. Like we didn't know that. And so these are like, they're just conveniently the simplest biological entity, and they're pretty safe to work with. And so there was a group of people that were working on them. And so it kind of like dovetails. We were learning about DNA and about microbiology and getting better tools and being able to see that these things were like a little particle with a genetic code that goes and replicates itself. Like we started to piece all that together, but phages weren't, used therapeutically in a standardized way too much. And I attribute that to kind of, I guess, two major things. One, we now know a personalized antimicrobial. So if you don't have the right one for the right bacteria, it's probably not going to work. It's like the Pokemon analogy. Like if you use the fire type against the water type, it's just not going to work. But you're blind. You don't know the types. So you're going to get lucky sometimes and not others. So we couldn't use them very well when we couldn't see. But the other big factor was antibiotics coming in and World War II, and that leading to just like the expansion, because they discovered antibiotics around the same time, like 1928, I think. And so antibiotics are not personalized in that way. Like you do not have to be as lucky for them to work. You use them and it's like there's a few types, but there's not like hundreds and hundreds and thousands of types.
Right.
So if you're looking for something that's more likely to work without having to know what you're exactly dealing with, you're going to use antibiotics. And so that just usurped phages, but I don't think it's right to think of it as like usurping really, because you're going to use the thing that works better. And it worked better at that time, and it has for many decades. And it's only now that, things are shifting. We've used antibiotics for so long. It's in the world, it's in the water supply and in the ground. And so the microbes have evolved. And so now we probably do need to like move to the targeted line of attack, and phages are still there. We now have the tools to know which one to use. So it's very doable now for that reason. Yeah.
Yeah, and yeah, I mean it's the time no doubt is right because and we've done enough and we'll be getting into it as we talk more about the pharmaceuticalization or the proper clinical development of these and sort of discuss antimicrobial resistance. Yeah, I mean it's we got a problem on our hands, and phage definitely is going to play an important role, in my opinion, in the coming years, no doubt. I was wondering, Jessica, if you could talk a little bit about sort of the before we get into sort of the also the proper development or the large scale pharmaceuticals. I'm using the word not in a disparaging way, pharmaceuticalization, but so we ultimately can treat billions of people with phages one day. There has been this compassionate phage therapy segment, and you've written about it. You wrote a very interesting paper in April 2019. This is the Journal of Virus. It's called The Current State of Compassionate Phage Therapy, where you sort of talk about, what the status is and where you think we need to be going in terms of, everything from clinical development to pharmacokinetics and everything in between. And then also this other interesting paper. This was in Microbiology Australia, and it was talking about sourcing phages for compassionate use. It's quite interesting because obviously there's, I said at the beginning, to 10 to the 31st power of these on the planet. That's a decillion of them. So there's a lot of these viruses out there, everywhere, from Lake Baikal in Siberia to the Amazon River and everywhere in between. Talk a little bit about sort of what compassionate phage therapy means, what it's meant the last couple decades, let's say, and what the current sourcing process before we get to phage directory has been. I mean, it's not, hey, I'm just going to go pull some stuff out of the river and purify it and hope we get a hit. What does that mean, the compassionate sourcing component of this as well?
Yeah. So again, cool. You've actually read these papers. I didn't think anyone was reading them. So yeah, like this whole compassionate use phage therapy, I think so compassionate use exists for theoretically anything that's an unapproved drug. And a doctor, I had to learn this, I was in my PhD doing science, didn't know anything about this side of things. And compassionate use is kind of doctor driven, and it's them giving the FDA a phone call and explaining, we want to use this unapproved medicine that we think could work on this patient. Here's why nothing else works. Can we use it? And so people think the FDA blocks things, and of course they do, but when there's nothing working, there is this option that physicians can actually make this case. And so this has been the way that phage therapy has been administered for the last several years, many countries. And so I think there's actually 2,000 cases, compassionate use cases of phages that are published and documented. So there's many more than that out there because vastly not everyone is publishing the results of these. But so what happens is, because I mentioned it's a personalized treatment, the first thing you need, and most people that are doing compassionate use phage therapy, they are doing it this way, where they're checking first what bacteria is growing in the person. So, and specifically, because you have lots of bacteria growing in you at any time, you need to know which one's causing the disease. And so that's pretty much, it's hard to do. It comes from the physicians making a call about what's likely to be causing the disease. And then they have to go and culture it. So you might have been in the hospital where they say, oh, we're going to run cultures, or maybe you have a UTI, potentially UTI, and they're going to culture it. It's the same thing. So the hospital or the clinic, wherever the patient is, they have to get their bacteria cultured. So say they have pneumonia, they would have to maybe provide a sputum sample from the lung, like cough up sputum and get that cultured. Or you could get a blood culture if they have sepsis or something in the bloodstream. And so the hospital microbiology lab, because every hospital has a microbiology lab, they will check what bacteria that is. And then for phage therapy, so they would then kind of be stuck because there's no phages in the hospitals. Like the hospital microlabs, they have all the supplies of the antibiotics, and they have a pharmacy that has those. The pharmacies have no phages because there's no phage products on the market in most countries, other than pretty much Russia and Georgia, and that's pretty limited. So in the US, the pharmacist would have nothing to give them. They would just have the strain of bacteria growing on a petri dish. So at that point, a physician would have to be responsible to find a source of phages. And so they could, in the beginning, there was a couple of phage companies that they would pretty much, if some of them knew that these phage companies existed, they would call them up, email them and say, do you have a phage? I have a patient. And that company might decide to pause their current work on developing an actual phage product, doing clinical trials, and decide to help this doctor. And I'm told that that would cost them around $50,000 just in supplies to make a batch of the phage and figure out which one to use. And so, as you can imagine, companies can't really justify this easily. And so they only did it a few times for the PR, and that's all they could really do. So trying to paint the picture that in most cases it was like there's no source of phages, and so the doctor would not be able to proceed. Until the last few years, there started to be, it was Steffanie Strathdee's case that, right. So her husband, Tom Patterson, which I guess you've had an episode with her, that was the first time when academics started to be used as like almost a crowdsourcing of phages for these cases. And so Steffanie, being a professor, knew that academics respond to emails if you find their email in their papers, and she emailed a bunch that she could find and asked them to please receive the bacterial strain from her husband and see if they had phages. And so that was like a really point of ingenuity. And the fact that everybody listened and responded and tried was also really cool. And I don't think it could have been predicted that that would go that way either. And so that's when I came on the scene and had seen that this case had worked. And so we started to think maybe you could get other phage researchers to help. And I know we're going to pause before we get into phage directory, but yeah, so just to close out the compassionate use loop, once you do find a phage, because you have like finagled your way to find someone by email and like also got the culture and like literally put it in the mailbox and sent it to that lab. And then they would actually take the bacteria, grow the bacteria, go in their fridge and get all the different vials of different phages they've collected over the years. Maybe they've collected 5, maybe they've collected 200. Some labs have been at this for decades. And so they would then pretty much grow a sample of the bacteria, and then they would pipette a little bit of each phage on top of it. Very much just like the 1915, like seeing the plaque on the plate where the bacteria was dead. You would try to replicate that. And if you see any dead zones, you would be like, yes, vial 2A, like, let's use that one. And then you would email the doctor and say, we have one. And then they would be like, cool, what's next? And no one would know because this is how uncharted it is. And the labs would be worried that what if they get in trouble if this kills somebody? We don't know if it's safe. We don't know if we prepared it in a safe way. We're not a clinical lab. These are not clinical labs. And so their university might say, no, you can't participate in this. So then that would kill it, even though they found a phage for that person. Or the doctor might be nervous that their medical license is on the line. And how do they vet that this academic has made a clean phage preparation. No one knows what that means. So, but this is kind of how the early compassionate use cases were going. It's like very ad hoc, and it would just take the right combination of people willing to take the risk, probably driven by the patient's case being dire enough. And yeah, so if you were lucky, you could get this academic lab to, I would say more of the time, it would be the academic lab sends the phage that they found, so 2A that worked. They would send that to the company that you've convinced to like spend 50,000 on your patient, the phage therapy company. Like Adaptive Phage Therapeutics was one of them that was doing this. And so then they would do the final manufacturing, and it's, manufacturing sounds like a big thing, but making enough of that phage to give that patient and making it clean enough that it's not going to cause any reactions. And so then they would ship that to the hospital pharmacy, and the hospital pharmacy would have to receive that, put it in an IV bag, basically, and the doctor would watch the whole process, and that would be your compassionate use. So it takes forever, and it's only going to work if the patient is stable enough to wait long enough, several weeks to months for this, but also not stable enough that they can't qualify by the FDA to be dire enough to need this. So it's been hard. Yeah.
No, I appreciate you laying that out to see sort of where we've come from, and sort of even in as much as recent years, that it's been a tough process, and hence why it's exciting what you're doing on all these different fronts now. And I think this would be a great time to introduce some of your work at Phage Directory, and talk about sort of what you've been focusing on that side, because again, and we've been profiling on the show sort of the banking of other types of stuff, so everything from bone marrow to oncolytic viruses and things like that. Clearly, you're thinking about putting a very well structured, standardized process together here. In April 2021, Current Opinion in Biotechnology, Phage Biobank, Present Challenges and Future Perspectives. You outline a lot of those challenges and future perspectives, and then there's another follow-up in June 2022. This is in the Phage Journal about the Phage Australia relationship. And I was wondering if you could again take us into the genesis of Phage Directory, how you set it up, what was involved in setting up, and a little bit of some of the collaborations that you initiated through it.
Yeah. So yeah, taking us back to 2017, I'm in my last semester of my PhD. I feel like a kid at the time, just a PhD student, and I'm told, lowest common denominator by other scientists, like you have no business starting initiatives. And that has sat with me for years because it's wrong. But, so I was there with Jon. We decided we're going to help facilitate compassionate use phage therapy, basically, by having a website that people could sign up if they have phages in their lab, and then which phages do they have. So, I have Campylobacter phages, you have E. coli phages, and then a way that you could contact all of the people who'd signed up as labs if you're a doctor. And so if you're a doctor, you reach out to us, we would reach out to the entire network and find a phage for your patient. That's kind of what we, proposed. We built a website. Jon built the website, and we went on Twitter, and we also started just emailing all the phage researchers that I and my professor at the time, Christine, knew in the phage world, and said, like, hey, do you want to be on this list? Like, we don't know if it will work, but you could help, because actually doctors are starting to want to use phages in the US and other places, and you could help. And so we did not think this would go anywhere. We actually were trying to save one specific patient that week, and that was Mallory Smith. And so she was a 25-year-old who had cystic fibrosis, and she had had a lung transplant, and then this infection took over the new lungs, and she was not doing well. So this was all around, actually, we weren't like, we're going to fix phage therapy. We were just going to help Mallory. But she died 2 days into this. And so and also the labs that we were talking to were like, what? Like she could have got phages and she died? Like, and so they wanted to help. And actually, Stat News interviewed us. And I didn't know what Stat News then. Now I do. And I can't believe they did. But they wrote like this beautiful article about Mallory. And then at the end, like, luckily there's a phage directory now. And so we were like, oh man, this 4-day project, we have to keep going with it. And so, I'm still working on my thesis, supposed to be working on my thesis. But this became so much more engrossing and fun. And, every day Jon was like, okay, write me a paragraph about what phages are, write me a paragraph about what compassionate use, like what the rules are, like who can help for the website. And I'm just like doing this on the side. And it was pretty crazy, but people kept signing up. And then doctors would reach out to us and they would ask for help. So this happened, I think, I don't remember, but a couple months into this, that there was like a real case and no one thought we were just like two kids with a website. They were like, oh yes, of course, the phage directory. I would like to request a phage. And so this is a doctor in France. And so we sent out an email to everybody on our list. I don't know how many we had at the time, probably like 20 labs. And some of them were like, yep, send me the strain. What should I do when I get it though? Like they didn't know what to do. And so we were just asking like Steffanie what they did. And Steffanie Strathdee was like a major mentor for us. She was our first call when we started this. And she was like, please make this happen. Like, I think she posted a tweet saying, please, could someone make a phage directory? She wanted like a bank of phages, naturally. And so this is our answer to that, almost like a virtual version of that and an on-demand kind of crowdsourced version. So you didn't have to raise any money to build a physical bank, basically. So gradually we would figure out, like, as the cases rolled in, just like ad hoc, what we're going to do. And we would link people when they didn't know what to do. We would find a lab that had done that thing and we'd just connect them. And eventually we kind of created a workflow that worked. And so it would be similar to what I described, that, a physician we decided early on, the physician had to be the one driving the case. A patient emailing us, it was hard for us to work with them without their physician driving it, because the FDA is not going to let them use it. They're going to have to say, go get your doctor. And then all these labs are going to do all this work, receiving a strain, looking for phages. They're going to burn out. And then there's not going to be a way to use those phages unless we have the doctor. So we kind of created that as a threshold. And then once that was in place, we'd make sure the doctor knew that they're responsible to talk to the FDA. They usually knew that. And then we made sure the labs knew that they're not going to be liable for the safety of the patient. The doctor is. And so even like they're not responsible by helping with a case. They're not saying we're giving you a pure, clean pharmaceutical grade phage.
Right.
We kind of separated that out. And that made it easy for labs to do the testing phase, which is really the best bottleneck for them to address. Is we need like thousands of phages tested very quickly. So we also didn't know in the beginning, like, would it be better to send phages to somewhere central? Like if a lab puts up their hand to help, we say, okay, send me your 20 phages. Or if we send out the strain to them. And like, it seems kind of obvious looking back, but it wasn't at the time. Like we kind of left it up to labs. It was optional. People would say, okay, I'll ship my phages. But in the end, it became wildly more easy to do shipping of the strain, because actually nobody really owns microbial strains that are in them. It's not considered part of it's not a sample in the way that like a tissue sample or like Henrietta Lacks, like a tumor sample,
Sure.
like
Sure.
a microbe is not yours. And so it's not like personally identifiable information if it's not got your name on it. So it's actually very easy for people to send. Nobody owns it. Nobody thinks that it's valuable. Versus phages, like the universities think, oh, I might make a drug one day. We can't let this out. And so taking the thing that's easy and not like people aren't worried about owning it and getting that thing shipped out, that was like the thing that we the nut that we cracked that really helped. And
Mhm.
so we started getting these things sent out. We would find labs would find hits, and then we would like find another third lab or company that would do the purification. And so we
Right.
just orchestrate that, lots of shipments. And then, yeah, we started getting, so there ended up being the first case where we actually treated a patient. And this was actually 2019. So it took 2 years of many like getting partial of the way there. But often the patient would die while we were trying to help them. Often patients would get better, and then their doctor would say, okay, never mind. We can't use a phage because they don't really need it. Or someone would get held up somewhere. Like the doctor would be like, you know what, this is too bureaucratic. My hospital is not going to work with me on this. I can't do it. Or sometimes it wouldn't find a phage at all. And sometimes nobody would put their hand up and say, I have one. And for a lot of species, that's the case. But so all these false starts, but we learned a lot. And then finally, 2019 was actually Jon Iredell and Amine Katami from Australia, who they reached out and said, we have a patient, 7-year-old girl. Her leg is getting amputated unless we treat this bone infection from this car accident. And so, do you have any Pseudomonas phages? We know we're in Australia, so probably no one will help us. Way too far, but like just checking. And and we sent that out, and I think that was our biggest response ever. We had 12 labs, and they were all from 12 different countries, and they all said like, "Oh yeah, I have Pseudomonas phages. I'll definitely help." And so we told Jon and Amine that, and they said, "Okay, we're ready. We're going to ship them out to these labs." And so surprises at each point because I didn't realize people would really, yeah, like help and want to ship to that many people, but they did. And so there ended up being a phage that was identified in Israel, and that was shipped to the US for
Mhm.
purification and then shipped to Australia for treatment. The Australians had to get all their ducks in a row, regulatory side, to get this to be allowed because it was the first time they were doing that outside a clinical trial in Australia. And they got everything ready so that the entire process took 6 weeks from them talking to us and like them treating this girl. So that was like lightning speed in phage world.
Right.
And then she actually she did not have to get her leg amputated. They resolved the infection without that because of
Mm.
the addition of the phages. So that was a huge win. That was the first full circle win. And from that, that led to this amazing collaboration that led to us moving to Australia for years and helping them establish a phage therapy center there because they got grant funding based on the success of this case and their other work, and were able to like bring us out. And we joined them for a time, and we just got back last year. So yeah, pretty amazing.
Yeah, and again, you say lightning speed, and I want the audience to appreciate, again, where we've come from to sort of what's been going on in the compassionate use world, and again where we'll be going in a little bit when we come to one of your really major papers from just last couple of weeks ago. One place though I wanted to stop in between, just because I think it's a cool concept, is you published in 2023 in the European Respiratory Journal on this citizen phage library project. And I thought this was really interesting. I just, a couple months ago, we had Craig Venter on the show, and, he was talking about his journey all around the world, and every couple hundred miles they pick up seawater, and 10 billion phage here, 10 billion phage there. This isn't, some obscure flower in the middle of the rainforest that we got to, hunt down and figure out. There's a lot of them, as we talked earlier, and I'd love to see if you can say a couple words about this, because clearly, we talk about citizen science and all these, the potential for, common folk like me getting involved, but, whether we're talking about, River X or the one I'm a couple blocks from a major river here in Philadelphia. What is the potential for the average citizen like myself to get involved in collecting and characterizing and sending or whatever phage resources, and what's that all about? Let's take us into this project.
Yeah, no, I love that you brought that up. Yes, so that is a project driven by Ben Temperton and this Citizen Phage Library project in the UK. And so Ben is one of the labs that has, I don't know when he started responding to our phage alerts, but he has been like a big participant in that. And he started this program. But before that, he had his research lab just isolating a lot of phages. They just got really good at doing that. And they really believe in like this should be a community. This is like a brilliant place for citizen scientists, like just anybody who's not doesn't have to be going to school for science, but they want to do science. And so he started that initiative and kind of just, I guess, graciously brought us into it. And we call him though. But they figured out kind of like a nice streamlined way to go and collect an environmental sample. So, like a water sample, sewage sample, like something from your rainwater, like soil, dog poop in your yard. And they made this little kit that you can actually order from them. And it's pretty much just like a tube with a filter. And because they found that, well, we have to filter the water to get the phage out of it. And if you filter it right away when it's collected, it works much better than not. So they created this little kit and they will send it out to people that want to help. And when like what help means is you would receive this in the mail, go and fill that little vial with whatever gross source, like literally anything could have a phage in it. And as you mentioned, it's not that rare. It's not like you have to go to the rainforest. It's like it's actually very, very common. And this is like Pokemon collecting. They're going to be different in every different place in the world. So
Right.
that's another reason it lends it to citizen science and crowdsourcing is like diversity is on our side. And it's actually easy and cheap on a one by one basis. It's just expensive to go to all around the world. So let's use the people that live in the places. And so you can go and say you go to your garden, get your soil sample, add some water to it, do the filter, and like you ship it back to the citizen science lab and they will have their microbiologists, who are many of them are students, and they are going to isolate phages from that sample. And so they're going to grow bacteria, put it on a petri dish, exact same 1915 experiment, and they're going to put some of your water on there. And
Hmm.
if they see a plaque, like a little clear zone, they'll take it and put it in a new vial, and that'll be a phage X, and they will do a few more checks to make sure that's really a phage, not just some other thing that killed bacteria.
Hmm.
And they will sequence the genome and figure out like which phage it is and is it new to science. And often every time you go look, it's pretty much you're going to find something new, which is kind of amazing. Like it's not, because as you said, there's 10 to the 31 of these out there. Like we don't know exactly how many different ones make up that 10 to the 31, but it the fact that we keep going and finding new ones every time we look it kind of suggests that there's a very high diversity. And so chances are you're going to find a new one, and then you, the citizen who isolated that, are going to get to name it. And that will be its name for all time in the scientific record. So unlike mine, NCTC 12673, which came from the National Culture Collection of the UK,
Hmm.
actually. That's why it has its name. But like I dreamed of being able to name a phage like that. And I still have never found my own and named it. I just use other people's. But anybody can. So you can go to this website and yeah, take part. And then Ben Temperton, whose lab it is that's doing this, he actually leverages all those phages in response to our phage alerts. And so that's the other way you can actually help with the patients too, is if your phage kills the patient's strain, doesn't matter who isolated it, it is now like going to go forward and maybe be helpful.
Yeah, I'm getting involved in that. It's just
Yeah.
again, it's cool citizen science, and I love hearing about projects like that. So no, that's really interesting. Um, so yeah, I mean, this brings us to a really important paper, and you just published this a couple weeks ago, Journal of Clinical Investigation, Bacteriophage Therapy for Multidrug-Resistant Infections, Current Technologies and Therapeutic Approaches. And we'll put a link to this paper in the bio of the show because it's an encyclopedic set of knowledge around everything that we need to be doing moving forward, especially in this era of multi-drug resistance. And it's clearly, we've done enough shows on this, and it's important that we got a problem on our hands in terms of the so-called silent pandemic. It ain't that silent, and this is a
Yeah.
problem that's with us now. It's coming very strongly. I don't, Jessica, I'm going to go into all this paper, but I just thought there's a couple of things I'd want to touch on because they're just interesting to me, but I think they make for an interesting discussion. And again, we'll put the link here, a very complex sort of pharmaceuticalization of how this should be happening. One of the interesting areas is a segment in this paper on, when we get to personalized phage therapy, sort of how we decide ultimately whether we do a fixed phage versus a cocktail, because there's a lot of them out there doing different things. And as you pointed out in your dissertation with 12673, very unique mechanisms of action. In this case, you were working on, okay, this virus likes to knock out the flagella, and that's cool. Antibiotics don't do that. Our current antibiotics only do a couple things, cell wall synthesis and protein synthesis, and then we're done. No doubt there's a lot of really cool mechanisms here. And I'd love it, to sort of just go into sort of your thoughts on, once we do have these amazing banks, strategies for putting together proper cocktails. Clearly, these are living items, and phase one and phase two might work totally differently, but have really great synergies. Take us through some of your thoughts in that area.
Yeah. Yeah. It's definitely just orders of magnitude more complicated in terms of how many different things could be happening at the cellular level when you're using these. Yeah. So instead of a small molecule antibiotic, they're all called small molecules because they're small and they're very simple. They have one mechanism generally, and phages are they have hundreds of genes each. Many of them do. And we don't know what most of them do, most of those genes. And the phage itself has many different things it's going to do to a given cell. And it's going to cause the cell to evolve in different ways. And we don't know what those ways are. And if we're using these phages as we're using a library approach where we're going to isolate a whole bunch of them and use whatever works. So we're being driven by what the patient has, which I think is the right way to go if you want effectiveness. And I think people would agree that, yeah, it's going to be more effective if you use a phage that definitely works on that bug, of course. But if you're driven by that, then you might be using a different phage every time. And so you have no time to study each phage, because each phage takes, I did a PhD on that one phage and I scratched the surface of it. An entire 5 or 6 year PhD would not be enough, not at all, not even close to enough to study one phage fully. And so we just can't know enough to that degree about all of them. But it's only in using all of them that we can really make them work right now. So that's the conundrum. The good thing is we don't have to, as long as we're reasonably open to some unknown genes being in there and some unknown things happening, we don't have to know everything about the phage to make it reasonably safe. And so you could start to bucket the phages into groups where okay, this one's kind of flagella specific and this one's kind of lipid specific. So the lipid on the outside of the bacteria is what matters. And people have done a lot of that grouping so that now when you get the genome sequence of a phage, maybe it's an unknown phage, you can at least compare it computationally to all the ones that have been known before. And you can start to guess at what group it might fall into. And so maybe you think, okay, this one that has a flagella specific mechanism, let's use that one in combination with this one that has a lipid specific mechanism, because then they're not the same mechanism. And so then the idea is, yeah, you're going to have this cocktail effect. You're not going to be redundant. So in terms of like making it work better, like that's one of the ways that you might choose phages is intentionally picking these like non-redundant ones. So that's great. It doesn't, get at, there's still potentially like different mysterious genes in each of those. And I guess right now the community generally, the sense is that that's okay. Phages are considered safe. And I think that statement is the underpinning of why this entire thing works. Because if they weren't given this like baseline assumption of safety, we wouldn't be able to use them in the way that we do and be okay with them being a bit mysterious. But overwhelmingly, use of phages has not led to poor safety outcomes. And so even the FDA has characterized some phages as generally regarded as safe or GRAS. They've gotten GRAS status. So that's not the case for any antibiotic or anything like that. So we have that advantage, but certainly when you're going to go and choose which phages, I would say phage choice is a whole field of science at this point. And that's a little bit what we went into in the review is like, we're trying to document how people are doing phage therapy. And a lot of that is compassionate still. And so that means a lot of it is also, it's not standardized because there's no one standardizing compassionate use work other than the safety aspect. And so yeah, it's happening in a lot of different ways. Some people are making cocktails very intentionally. Some people are making them, like in Australia, where I was the last couple of years, we were making them in a very patient-targeted way. So they were like, we're going to make a cocktail of 3 phages, 3 because that seems like a solid number, but not too many that it's too hard to make. But, let's do 3 that are going to work on that patient strain. Well, we didn't check if they were non-redundant. We didn't really care if they were. And we did check if they had synergy or antagonism with each other. We would combine them and see if that still worked. If it worked less well, then we would probably not use that combo. But we didn't see that much. So yeah, we were kind of using very low levels of characterized phages, but very targeted to the patient strain. But we were using cocktails whenever possible. Versus there's the other side of the spectrum where you could use fixed cocktails where it's like, and this is what companies are doing. They'll pick like for a given bacterial problem, like Pseudomonas aeruginosa, they'll pick 3 phages that are going to work on the most geographically, like just the most strains possible of that species. So they'll collect thousands of isolates from nature or from the CDC or wherever they can get. They will find the 3 phages that give you the broadest spectrum the most consistently. And then they'll only make those phages. And then if a patient comes along, they get that fixed cocktail. And that has its advantages, obviously, and it's going to be the way that it goes if it gets scaled up, of course. But I would say that's kind of the other side of the spectrum at this point.
I appreciate the sort of the generally recognized as safe component too, because we don't think about it as much as we should. That here we have organisms that are quite ancient and that we've co-evolved and so forth. Kind of like the microbiome stuff, right? I mean, here with the microbiome therapies, that they're extremely complex cocktails. We don't need to have 100% standardization of bacteria 1 versus there's a little magic, let's just say, in the way nature combines this stuff, and it's I can appreciate that aspect of it. As far as some of the again, if the sky opened up and $1 trillion landed in your bank account tomorrow, are there certain clinical indications that kind of excite you? I mean, I know that it's quite broad, and in the paper you mentioned potential sort of topical things like diabetic foot ulcers, joint infections from prostheses, urinary tract infections, which may be a little easier to dose. What's kind of your vision there? And obviously, with $1 trillion, you go in a lot of directions. But what are the things that excite you in terms of some of the shorter term clinical development you'd like to see?
Yeah, I think the one that really there's a lot of people using phages in the lung, and I think especially for chronic diseases like cystic fibrosis, there's like, I think that's a great place for phages, and they're using them there. But in terms of what the unmet need is, like, I just feel like the amount of amputations that are happening because of bone infections, the amount of repeat surgeries because of your hip replacement, like whatever
Yep.
metal you have anywhere in your body, like that's just going to grow bacteria, and then they're going to have to keep doing surgery. Like people get like 17 surgeries. I met this guy who had 17 surgeries, and like he eventually got phage therapy and then didn't need to keep getting those surgeries. Like I I feel like that's a very satisfying place to put efforts, because that's also, there's just no way to treat bacteria that are growing in biofilms on metal inside the body. Like that's not a natural uh, thing for, I mean, I guess it's natural because the bacteria are growing in their nature, but it's very hard to treat with antibiotics, and it's always going to be hard to treat with antibiotics. And
Mhm.
phages can really get in there, embed in there. They have biofilm breaking enzymes, and they can kind of replicate each new bacteria they kill, they can replicate a little bit more and just like get in there until it's over.
Mhm.
And so I would probably focus on those, and many people are. It's just hard. But yeah, like that's it just seems so treatable. And diabetic foot infections, like
Mhm.
I don't know if you've ever delved into that, but
Yeah.
that just I didn't realize that like they consider like once you like people have diabetes, like their limbs, like the circulation just gets very, very poor. And then
Right.
it doesn't even matter if an antibiotic could work, it doesn't get there because there's no blood flow. And so you just get these non-healing wounds, and then it's just like a matter of time. Like the doctors just go, "Oh, you're going to get your amputation, most likely at this point." And once you get amputation, they talk about 5-year survival, and it's like it's almost like a cancer diagnosis, but people don't consider it like that. But it's like pretty much sure that uh, once you get a lower limb amputation from diabetes, like
Mhm.
the clock is ticking. Like that's
Right.
and that's from an infection. So yeah, that's kind of one area that I think they could do a lot.
Cool. And one other thing, Jessica, while we're in this paper, and then I'll move on to a couple of things. But there is one little section on sort of the phage antibiotic synergy concept, and there's a really cool researcher at the Naval Research Labs, Dr. Biswas, that he's done a lot of work over the years. He leads the phage science division there on again, combinations and ultimately, is there a way that phage can maybe extend the life of, let's just say, the existing antibiotics, which aren't doing that well, but may have some synergy there. And I don't, I know this is kind of an odd question, but thoughts on sort of, I mean, I don't know if when you have something that's vancomycin resistant, whether it's 100% vancomycin resistant or 50%, whatever, whether you see any interesting work in sort of the combinations with antibiotics, and any interesting research that you've read along the way as you've
Yeah.
been doing this amazing work. Again, sorry, long-winded question there, but
Oh,
your
love it.
thoughts. Yeah.
Yeah. Yeah. No, I think that's the way that we need to be going. I'm not one of the proponents of just like replacing antibiotics with phages. I think they need to be combined strategically. And there is such cool work, especially coming out of Yale, Paul Turner and Ben Chan's group. So they have started, instead of like other people are using phage cocktails, right? But they're using one phage at a time and getting really clear on what happens to the bug after.
Mhm.
And can they strategically pick phages that are going to target a bacterial either receptor or protein that is involved in antibiotic resistance? So, for example, a multi-drug efflux pump, that's like one of the mechanisms that bacteria will just get any antibiotic that comes in, it will just shoot it out. It's a pump. And so it recognizes antibiotics and it pumps them out. And they had the idea to find a phage that needs the multi-drug efflux pump as a receptor to get in.
Mhm.
And so it's going to kill anything that has that pump. And then the bacteria, they're always going to evolve too. and they're going to evolve to drop that pump. And they had a hypothesis that this would happen, and then it actually did happen when they checked in the lab. And so this phage OMCO1, which I actually work with in the lab now, it will have this effect. And so you get resistance, but now it's more treatable because it doesn't have a pump. So you can hit it with regular antibiotics again. So if you know something about each bacteriophage and you choose them based on that, and we do know a lot about the resistance mechanisms of a lot of antibiotics. Like, luckily, we know that quite well. There's not that many antibiotic classes. And so we can go, okay, in what ways would, like, is there something on the surface of the bug that a phage could target? And I think that's like a very promising area to use them. And like they were able to show that in a patient as well. And it actually like treated the infection, and they combined it specifically with that antibiotic, and it worked as it was supposed to. So pretty cool. It's just a matter of developing that atlas of what are the mechanisms we need phages for, and then kind of probably just turning it to the community to go find those phages. But you can find a phage on any. The other cool thing when you're finding a phage is like which bait you use, like which bacteria do you use as bait, because you can be strategic about that. And that hasn't been done as much as people just finding phages on like any given lab strain. Like that's what most of the phages that we have are from. But like if we started shifting to like strategically like, okay, we're going to use this really bad bug that we can't kill with antibiotics, then that's going to be the bait. And then you go phage hunting, and you don't need to send that bad strain out. You just need to use that when you receive all the phages. So there's a lot that could be done there, I think, being strategic about that.
Very cool. Very cool. Just we talked a lot about human health, but clearly there's opportunities all over the place beyond that sector, and it could be food safety. You mentioned the experiment, I think, in India in terms of wastewater treatment. You mentioned biofilms before, and I remember this ridiculous $5 trillion number of the economic cost to all the sewers and the ships and everything else where the biofilms impact those surfaces and cause all sorts of problems. What else gets you excited in terms of applications of where we're headed with phage, looking out over the horizon beyond human health?
Yeah. It's pretty, I'm excited about what's happening, definitely for livestock feed. And I went to a conference in Colombia last year, and there's a whole contingent of people in Latin America that are starting to use phages. And I guess their regulators are really amenable that even like European and Canadian companies are looking at doing their trials down in like in South America. And so, yeah, there's some really cool stuff happening with like anti-salmonella phages, and that you would feed that to chickens, and anything that's going to this is exciting to me because it like fixes the actual root problem of antibiotic resistance.
Yep.
If you think of it as coming from livestock and coming from like antibiotics being in our water supply and runoff, that's because we're feeding them to animals. And so we can do what we want in the human clinic, but that's like a band-aid. Like that's like the final thing. Like the root is really to get antibiotics out of our food and water supply. So if you can start like treating these animals and giving them phages, then you can comply with the whole no antibiotics in meat rules that are starting to come out, fortunately. So and that would have a big outsized difference. I know it's been very hard for like I know a Canadian company, Cytophage, they are trying to do this for chickens, and they've had to run all their trials outside of Canada because it's been like extremely overregulated is the sense. And I don't know, even though I'm Canadian, I'm not following the specifics on the regulatory side of things there, but I still really want this to move forward in my country, but they're having to do their trials and probably only sell their products like out in Asia and because I guess the Canadian government wants even agriculture phage to be like at the same level of manufacturing as human
Hmm.
products. And because phages are still seen as a little bit like unknown and risky, I think. And so if we start to see different regulations that carve out like a phage specific pathway
Hmm.
across the board, that should shift things. And then hopefully, yeah, phages can be dumped in the fish food and the chicken food. And obviously, we got to check that that's not having unintended effects. But right now, we're dumping antibiotics in there. So, yeah.
Yeah. Well, we unfortunately been killing a lot of chickens and cows and everything else from all the spillover infections. So yeah, I mean it's going to be an important tool. I look
Yeah.
forward to seeing how that whole agra, defense agra prep area evolves. Yeah, no, it's really interesting. Just what else should we be on the lookout for in 2025 as it pertains to phage directory, to your work at Stanford, to conferences, talks, anything else on in the coming months that we should be aware, or anything else that I missed while we have you today,
Yeah.
please?
Yeah. Well, one thing I'm really getting into lately, especially after joining Stanford, because it's a like such an engineering focused school, we're starting to
Hmm.
look at like to what extent, kind of harkening back to the use of phages in the 50s, 60s, 70s to understand DNA and to use them as biotechnological tools.
Hmm.
In addition to the phage therapy stuff, I'm getting really excited about pushing the limits of what we can use phages for from an engineering side. And
Okay.
we have like so this actually opens up phages to help treat genetic diseases and like cancer as well, and so not just bacteria diseases, and so that you can use them as a gene delivery vector and a peptide delivery vector theoretically.
Okay.
And
Hmm.
so this is kind of like the exciting work that I'm looking toward because as I'm hearing from around Stanford, there's a lot of people doing gene therapy and a lot of people trying to put CRISPR, into a certain cell type, and they're using viral vectors, but they're using human viruses to deliver that gene. And
Mm.
we're starting to find that phages can be shuttling stuff into human cells if you engineer them right. And if you use the ones that actually naturally are taken up by certain cell types. So I'm starting to think like instead of prokaryotes only, like bacteria, like eukaryotes, and like how do we get phages to do our bidding in that context? And I think because we're kind of hitting a wall, the gene therapy, peptide therapy field is, as I understand it, hitting a wall with what they can deliver, how big of something they can put in. Like you're so limited by like genes, you can only bring in a short gene. And so, and CRISPRs, like they take up a lot of room inside a virus, but phages
Right.
are way bigger viruses. And so I think this unlock could really help us in a lot of ways. And like, even if it takes a long time for the phage therapy field to keep advancing, like we can go back to what phages really helped us do in the past, which is like understand like basic stuff about bio and like bioengineering and actually like return to that, but then apply it to this like new therapeutic area that's like hitting a wall. So that's going to be what I'm looking at doing and figuring out how I can expand phage directory to kind of involve more of the eukaryotic people and figure out like I want people to, you know, reach out and say like, okay, we want to deliver this cancer peptide, but we can't put it on a human virus because the human virus just gets shuttled to the liver. But like we could put
Right.
it on this like Trojan horse phage that's not very immunogenic and can actually, if it can get there, would be great. So that's the new frontier for me, and we'll see how that goes. And on the other question on what conferences and cool stuff coming up, there's I'm co-organizing a conference, which if people are interested, like I know this isn't like your phage audience, but we've had people from the community come to this conference. It's called the Evergreen Phage Meeting, and it's
Okay.
it's been running for 50 years. So it's run by this woman, Betty Cutter, who she's been a scientist She got her PhD 50 years exactly before I got mine. So it was pretty cool. So I've known her for my whole scientific career, and she's been running this conference every other year for 50 years. And so it's going to be in Knoxville, Tennessee in August. And it's like phage summer camp, basically. And we got phage biology, phage engineering, phage therapy, and that'll be a week in, yeah, Knoxville.
Cool. Very cool. Yeah, no, again, this is an area that excites me. It's great to hear your passion and see you leading the charge now and moving these programs forward. Maybe 100 years after the fact, but yeah, I mean, it's definitely 2025. We need this in a major way. So I'm again excited for you. I wish you and your team there the best with all of it as you continue to progress as you are doing. But really impressive work. Again, for everybody that is going to be listening to this episode of the show on the various podcast networks or watching on the YouTube channel. Again, you've been spending time with Dr. Jessica Sacher, co-founder of the Phage Directory, staff scientist, Stanford University School of Medicine. Check out the Capsid Tail, as well as the Citizen Phage Library, and we'll put links to that in the bio notes of the show, and also check out the Evergreen Phage Lab and the Knoxville conference. Yeah, Jessica, it's really awesome having you on the show and going into this topic with you. I really, wish you the best with all of it. Thank you for all you do on this front. And again, we say on our show, thanks for helping to create a better tomorrow for many people out there via this type of work. It's so very important, and really, really appreciate you and you spending time talking about it today.
Thank you so much. This has been just a delight to have this conversation. So very
Really,
excited to get the message out.
really great having you, and we'll get it out there for you. Great having you today, Jessica.
Awesome.